Good morning, everyone, and bonjour. Unfortunately, I was hoping that we would be in Quebec City today, and I would have enjoyed a lovely extended weekend in a gorgeous city in French Canada that I've never visited. But the pandemic situation has made that impossible. But it really is my pleasure to be here today, and I'm very grateful to the meeting organizers for inviting me to give this plenary talk. The talk today is antiretroviral therapy for treatment and prevention, beyond daily oral therapy, a new frontier. These are my disclosures within the past 12 months. Today, I'm going to review the current recommendations for combination antiretroviral therapy in HIV-infected individuals and why we need alternatives to daily oral therapy. I'm also going to present the same type of discussion for HIV prevention. I'll then segue into long-acting injectables antiretrovirals for treatment and prevention, detailing long-acting cabotegravir and rilpivirine for the treatment of HIV infection, as well as long-acting cabotegravir for pre-exposure prophylaxis. Following that, I will talk about the exciting new field of broadly neutralizing antibodies, which are being developed both for treatment and the prevention of HIV infection. And finally, I will conclude with a discussion of islatravir, otherwise known as MK8591 or EFDA. This is a compound being developed by Merck that I've been fortunate to work with in both the PrEP and PEP animal model. So since I'm in Canada, it's critical that I include the Canadian guidelines and ici est la recommandation pour le traitement de VIH, Québec. And that's the end of my French. And of course, it's a busy slide, but basically it says what we all know. We use 2 nukes and a third drug, be it an NNRTI, an integrase inhibitor, or a protease inhibitor. And in the province of Quebec, I think there's a fair amount of choice for the individual practitioner to choose from these nuke combinations as well as the third agent. And here, on the other side of Canada, are more guidelines, I think, that are a little more rigid in the preferred regimens, in that they really do prefer or recommend the integrase inhibitors, since I think most people are using integrase inhibitors these days, and fixed dose combinations of bictegravir, TAF-FTC, otherwise known as Biktarvy, or dolutegravir, bacavir, and 3TC. Those are the most common. Interestingly, they also do mention dual therapy for patients with viral loads less than 500,000, specifically dolutegravir and 3TC. That has also entered the US guidelines. And I think the guidelines now are pretty clear that most people can be treated with convenient 1 pill, once a day therapy. Now, what is this based on? And here I have some data from studies performed by Gilead in comparing bictegravir FTC and tenofovir alafenamide to dolutegravir, bacavir, and 3TC. And you can see quite nicely in both studies where here it's a dolutegravir, abacavir, 3TC, and here dolutegravir, FTC, and TAF. So Biktarvy is basically being compared to 2 different dolutegravir regimens with very high percentages of patients who are below 50 copies of RNA per ml at 48 weeks, 96 weeks, and even out to 144 weeks, which is extremely exciting data. And when we look at the reason for the drop-off in both studies, we can see that most of the patients don't have data as opposed to true virologic failure, which would be shown in this part of the slide. And in these studies, when they look at patients at week 144, whether treated with Biktarvy or dolutegravir-based therapy, there's essentially no patients who have treatment-associated emergence of resistance. So it would appear that we have really perfected one pill once a day options for HIV therapy. So why would we possibly need new treatments? Well, this is why. These are data from the US CDC from 2016 showing the HIV care continuum. And in the United States, it's estimated that approximately 86% of people who are HIV positive are diagnosed. And of that, approximately 78% of those diagnosed are linked to care within a month. And as you can see, from that 78%, the 64% receive care, and a terrible 49% are actually retained in care. And of all the patients who are HIV infected, about half of them are suppressed virologically. So despite the fact that we have more or less perfected daily oral therapy for treatment, we have a long way to go, and we need alternatives to our current treatment strategy. So let's move on to HIV prevention. And here are the Canadian guidelines that appeared in 2017 in the Canadian Medical Journal. And basically, PrEP with TDF-FTC taken orally is an effective strategy for reducing the risk of HIV infection. It does also say that in the population where it's been tested, an on-demand regimen may also be considered. And more recently, this asterisk here just highlights the findings of the Discover study, where TAF-FTC was found to be non-inferior to TDF-FTC in a large cohort of sexually active men who have sex with men and transgender women. And it was noted that the TAF-FTC group had less bone toxicity. But it's also clear in the Canadian guidelines, current Canadian guidelines, that there's a big difference in cost. And that raises the question whether it is indeed worth treating large numbers of patients with TAF-FTC as opposed to TDF-FTC. And I think that's a question that will continue to be answered over time. But what do we know about effective PrEP? Well, this is an old slide. It's from the AVAC group. And it shows some very important, I think, points about HIV prevention and also HIV treatment. So on the x-axis are the percent of patients who have detectable drug levels in their blood. And on the y-axis is the effectiveness of a particular regimen. And you can see quite clearly that there's a very high relationship, a good relationship between having drug in your system and getting the desired effect. So in studies where there was a very high effectiveness of PrEP, most of the patients had drug in their system. And the studies where there was no effect, there were a very low percentage of patients who had drug in their system. And in the studies that had intermediate effects, an intermediate number of patients had a drug in their system. And this really is something we've known about HIV treatment and now HIV prevention for a long time. Drugs work when patients take them. And it's pretty clear that the treatment data I just showed you, a lot of patients aren't taking drug. And we also know now that a lot of patients are not taking PrEP. So this is a paper that came out of San Francisco Department of Health primary care clinics. And in their series, most PrEP adopters stop using drug in 6 to 12 months. And this is an issue of what the field now calls PrEP persistence. And actually, the mean time of use currently is 8.2 months, with Blacks, trans females, and people who inject drugs being at highest risk to discontinue PrEP. And the factors are complex. There are individual factors, such as the perception of reduced risk, side effects of the medication, and I think most commonly, there's difficulty with adherence to daily pill taking. And then there are contextual factors, such as the ability to afford the medication and the healthcare associated with PrEP use. And this is probably more of a US problem than anywhere else, because we have a relatively broken healthcare system, as is evident from our COVID-19 problem. And also the healthcare relationship between the clinic, the provider, and the pharmacy with the patient. And this is critical because there are segments of our population in the US, and I assume the same is true in Canada, where the relationship with the healthcare system is less than ideal. And we can never forget about the stigma of using antiretroviral medications. People who use antiretroviral medications are thought to have HIV infection. So this is an important issue and something that really stymies effectiveness of PrEP. And also, it's also thought that people who take PrEP are somehow more promiscuous than people who don't. And there's additional stigma there as well. So if we look at these factors, both in the treatment space and the prevention space, we can see quite clearly that we need to go beyond daily oral therapy. And as I said, I don't see much ability to improve what we already have. So therefore, we have to start thinking about alternative drug delivery systems that can go from taking pills every day to perhaps giving medications in a different way with different dosing regimens that are further apart, so it takes away issues around adherence, etc. So the first advance I'm going to talk about is intramuscular long-acting cabotegravir and rilpivirine for the treatment of HIV infection. And we can say quite clearly, and I can say this with almost certainty, that based on the 2B and phase 3 randomized clinical trials, that this two-drug maintenance therapy will likely be the first novel delivery modality approved to treat HIV infection. And it came up for approval here in the US, and it did not get approved, not because it's ineffective, but because of issues surrounding production. So it's a CMC issue, and that hopefully will be solved in a relatively near future. Now, in these studies, there was enormous benefit of once a month to every 2 month injections. Obviously, reduced dosing frequency and adherence to visits becomes more critical than daily pill taking. And that's something that the healthcare system can actually observe as opposed to trying to assess when patients are at home. Now, the other very important aspect of long-acting therapy with this particular combination is the quality of life questionnaires that were done during these studies. There were very high levels of satisfaction despite common injection site reactions in nearly all the subjects. And the patients appreciated convenience, the reduced risk of inadvertent disclosure of having oral medications in the home, and also for many of them, the daily reminder of their HIV status when having to take pills every day. Participants in these studies also felt that the stigma of having HIV infection was reduced, but particularly in the patients who were on the monthly treatments, they wanted longer intervals between treatments. So let me first talk about the 2B study. This is called LATTE-2, and basically it took naive patients, it induced them with triple combination therapy, and if they were suppressed, they went into a challenge with oral rilpivirine to make sure they weren't allergic. And if they were not allergic to rilpivirine, they were randomized to one of three arms: a long-acting cabotegravir plus rilpivirine every 4 weeks at 400 milligrams and 600 milligrams, a long-acting cabotegravir at 600 milligrams and 900 milligrams every 2 months, or continuing daily oral therapy with pills. And here's the data up to 96 weeks as published by David Margolis and colleagues in The Lancet in 2017. And you can see quite clearly that the patients who go into the maintenance period have a very, very high rate of sustaining virologic suppression. And you can see that the patients who got monthly rilpivirine and cabotegravir had 87% of those subjects had viral loads that were undetectable at 96 weeks, as opposed to 94% in the every 8-week arm and 84% in the oral therapy arm. Now, because there were some virologic non-responders in the every 8-week arm, and this was thought to be perhaps related to decreasing levels of the rilpivirine, which has kind of borderline pharmacokinetics when given every 8 weeks, the every 4-week regimen was taken initially into phase 3. And these studies were called FLAIR and ATLAS. The FLAIR study recruited naive patients, induced them with triple combination therapy, and then randomized them to either oral drug to make sure they weren't allergic to rilpivirine, followed by Q4 week 400, 900 rilpivirine. Notice the rilpivirine dose is higher than the 600 milligrams that was tested in MATE-2. And the other arm was daily oral therapy. In the ATLAS study, patients were randomized to receive, this is a switch study where patients were switched first to oral drug to make sure they weren't allergic to rilpivirine, and then treated monthly with cabotegravir and rilpivirine, or continued on daily antiretroviral therapy that they came into the study on. Now, the exciting part of this, and I'll show you the data, the results of FLAIR and ATLAS, is that as LATTE-2 matured and showed that the Q8-week arm was performing much better at 96 weeks than it did at 48 weeks, this study was extended into what was called ATLAS-2M, where the regimen is being given every 2 months instead of every month. And this phase 3 study will likely form the basis for an approval for every 8 weeks. But right now, it's likely that the intramuscular combination will be approved first for being given every 4 weeks as maintenance. So here are the results that were presented at CROI in 2019. I apologize for the slide, but because of the current work situation, I had to take this off the CROI oral abstract website, and I couldn't get a better slide, but you can see quite nicely that in the FLAIR, which was the naive study, very high rates of undetectability at 48 weeks. And the primary endpoint was virologic failure, and you can see non-inferiority. I apologize again for the faded nature of this slide, but this is a result of having sequential slides on the same slide. But you can also see that the arms are non-inferior as far as looking at a secondary endpoint, the percent of patients who are less than 50. And here's ATLAS-2M. I keep going to ATLAS-2M because I participated in both the ATLAS and the ATLAS-2M study. But nevertheless, again, non-inferior as far as lack of virologic response at 48 weeks, and also non-inferior as far as less than 50 copies per ml. And you can see that there's a slight difference here between the long-acting and the oral therapy, because there were slightly more dropouts in the ATLAS, in the IM therapy arm compared to continuing oral therapy. And for most switch studies, this is not unusual. The patients who get the new treatments are often a little less tolerant of new adverse events than patients who you treat in studies where patients have never seen drug before. So just some caveats about the use of intramuscular rilpivirine and cabotegravir for treatment. Injection site reactions are the predominant adverse event. The pain is generally mild to moderate, but surprisingly, even though the pain can last for days, it rarely results in treatment discontinuation. And I told you earlier about the quality of life questionnaires, and the patients really did tolerate the treatment well. Very importantly, though, two-class resistance has been demonstrated. And some of this is probably due to poor adherence to the treatment regimen. Patients may have missed appointments. And also there were some patients who had low drug levels that may have been related to problems with injections. Now, very importantly, remember, these are viremic patients. And if they are lost to follow-up, they will likely get resistance because these injectable drugs have very long tails following injection. So replicating virus will likely see subtherapeutic levels of drug over a period of time. So if patients are going to be treated with this regimen, they really have to be carefully identified, selected, and followed. Let me now move into cabotegravir long-acting for prevention. This is work that was done at the Aaron Diamond AIDS Research Center in my group, along with David Ho's group. And this is just a summary of our first study where we challenged Rhesus macaques with SHIV-162P3 after treating them with monthly cabotegravir 50 mg per kilogram every 4 weeks, just to show you here that the drug and the monkeys, were challenged intrarectally with 50 TCID 50s of SHIV-162P3. And just to show you here that all the monkeys that were treated with cabotegravir, which was then known as GSK 744, were protected, and all the untreated monkeys became infected. And these are the viral loads. And just to sum up, most of the monkeys became infected on average after 2 challenges. So the treated animals had a 28-fold lower risk of infection with a very good confidence, 95% confidence interval. And by log rank, this is highly statistically significant. We also did an experiment where we gave a single dose of drug and then continued to challenge the monkeys until they became infected, and then checked drug levels. And what we found were that monkeys who had greater than 3 times the protein ingested IC90 of cabotegravir were completely protected. And animals who had 1x to 3x that concentration had about 97% protection. This 1 out of 22 doesn't translate into 97%, but you have to account for the fact that it takes on average 2 challenges to infect a monkey. And obviously, there's no statistically significant protection when there's less than 1x PAIC90 or no drug in the controls. So based on these preclinical studies, we did the first, I was the PI of the first phase 2A study of 800 milligrams long-acting CAB given intramuscularly every 12 weeks. And that was called the ECLAIR study. It was published in Lancet HIV in 2017. And I don't really have time to go into all the details. All I can tell you is that the drug was relatively well tolerated. It caused injection site reactions in nearly all subjects and all injections. And the pain was mild to moderate and would last approximately 5 days. What was disappointing, however, was that the modeling shown here in the orange dotted line was suggesting that concentrations of drug would be in excess of 4x PaIC90 throughout the every 12-week dosing interval. But in reality, shown in the green line, the drug levels fell significantly below that. And you can see that many of the patients fell way below that, even some down below the PaIC90. So it was quite clear that the hope for an every 12-week preventative was not going to happen with cabotegravir. And these are data from the HPTN 077 study that was done simultaneously with Rafi Landovitz as the principal investigator and published in PLOS Medicine 2018. And when they saw our data, they recruited a second cohort and gave the drug basically every 8 weeks after a 4-week loading. So patients at time 0 got 600 milligrams, 4 weeks later another 600 milligrams, and then 600 milligrams every 8 weeks. And you can see quite nicely that in the men in particular, the levels are quite high, and in the females in general as well. The drug levels are well above, on average, 4x PaIC90 and up to 8x PaIC90. And this is the dosing regimen that is now in phase 3 study of CAB-LA. There are 2 large studies, HPTN 083 with Rafi Landovitz at UCLA as the principal investigator. It's a double-blind, double-dummy comparison study of FTC-TDF, Truvada, in MSM and transgender women. The enrollment is really getting there, 4569 out of 5,000, and the study is powered for non-inferiority. There are 43 sites in the US, Peru, Brazil, Argentina, South Africa, Thailand, and Vietnam. However, because of the SARS-CoV-2 pandemic, enrollment is now frozen. Patients are coming in for their visits, but new patients are not being enrolled. And we don't know when this is going to actually be fully enrolled. But so far, the good news is that the study has not been stopped by a DSMB, suggesting that there's not a terrible imbalance in numbers of infections. There's also a study in high-risk cisgender women in sub-Saharan Africa, and the principal investigator is Dr. Delaney Moratiwi in Johannesburg, so South Africa, and it's enrolled 3,032 out of a planned 3,200, and it's powered for superiority, and it has 20 sites in 7 countries, all in sub-Saharan Africa. And again, enrollment is frozen, but patients are coming in for their visits. We were hoping for readouts of these studies in 2021, but we may have to wait longer because of the current pandemic. I'm now going to move on to probably one of the more exciting areas in HIV medicine, and that is the discovery and clinical development of broadly neutralizing monoclonal antibodies to HIV. It had been thought for a very long period of time that broadly neutralizing antibodies just didn't exist. And the reason it was thought is early on in the epidemic, the technology really didn't allow for the identification of such. But there have been advances in basic science that have allowed for enormous discovery. And what we've learned from cloning B cells of HIV-infected individuals is that there are very, very potent broadly neutralizing monoclonal antibodies to many domains of the HIV envelope. The patients from whom these antibodies have been isolated have generally been infected for very long periods of time. And these antibodies, or the B cells, are highly mutated from germline sequences. And I'm going to show you the single antibody cloning methods that have been used, and I'll show you some of the clinical trial data that has been done. So I am not a basic scientist, and I just wanted to show you people that what enabled the field starting in around 2009 and going up to the present was the number one recognition of A, the importance of memory B cells, two, the ability to either sort them or put single cells into a plate, three, either test supernatants for neutralization or sort the cells into 96 well plates and use high throughput sequencing, then get antibody clones and screen for neutralization. So there are two methods basically. This is a slightly older, this is the newer, and they've resulted in the discovery of many, many antibodies. And they're too numerous to count, but I do want to call your attention to a couple of the epitopes. There's the membrane proximal region right here, where GP120 and GP41 sit on the membrane. This is the antibody to this area is called 10E8, and it is a very potent and very broad antibody. And you'll see it come up in my discussion. This is a CD4 binding site, and you can see here that there are numerous antibodies that have been identified to the CD4 binding site. Perhaps the two most important are VRC01 out of the Vaccine Research Center, and 3BNC117, which was discovered by Michel Nussensweig's group. This is, there are also potent antibodies that bind to a carbohydrate moieties on the trimer of the viral envelope, and this can happen in the high mannose patch here, or the apex shown here in blue. And the apex, these PG antibodies are antibodies that were discovered in a collaboration between the group at UC San Diego, led by Dennis Burton, and IAVI. And really they refer to PG as protocol G antibodies, which were, and the protocol was basically drawing blood on patients who on the African continent who had been untreated and infected for long periods of time and looking for neutralizing antibodies. So as you can see, there are numerous antibodies, and they have many different, they bind to many different epitopes on the surface of the envelope trimer. Now, what is very exciting in the field, and this is, I always love to be wrong, because when people were talking about antibodies, I said to people in the field, I said, well, you know, antibodies have really short half-lives. So I don't see how you're going to do better than a drug that's being given every month, 2 months, or 3 months. But I love being wrong. And what the people who work in the antibody field were able to do was to engineer these antibodies for longer half-lives. And basically, these are called LS variants of the parental antibodies, and they take advantage of the fact that the immunoglobulins bind to the neonatal Fc receptor on endothelial cells, and these and the IgGs are endocytosed into the cell. And if it becomes unbound, it gets degraded. But you can actually manipulate the binding of IgG to the Fc receptor, to the neonatal Fc receptor, such that it doesn't unbind. And it actually gets recirculated back up to the surface and released into the plasma. And as you can see here, VRC01 has a half-life of 15 days, but the LS variant is 71 days. And their newer antibody, VRC07, which is also a CD4 binding site antibody, has a half-life of approximately 33 days, which is, again, very favorable for use, be it every month, 2 months, or even perhaps 3 months, depending on the dose. So here's some data of combination antibodies. This is a CD4 antibody, and this is a Apex antibody developed by the Nussensweig lab. And basically, when they gave these antibodies in combination once or 3 doses once a week, there were 3 patterns seen. Patients who had early rebound, shown in the black line. The colored lines are the drug levels or no response at all. And then patients who had very nice responses with very late rebounds. So you can already see that one of the problems with using combination monoclonal antibodies is that not all patients respond, very different from antiretroviral therapy, where most patients respond unless they have transmitted resistance. So when they went and looked at the susceptibility of the viruses pre-treatment and post-treatment, so what you're looking at here is that they're taking the viral envelopes and making pseudoviruses and looking at individual clones. So the patients who rebound late are susceptible at the beginning and don't have changes in susceptibility. The patients that have early rebound or no response are clearly far less susceptible, and some of their clones are completely resistant. Patients who responded but have late rebound seem to have the emergence of resistance, 3 out of 4. And then the patients who have early rebound and no response have a very rapid emergence of resistance in dual therapy. So here's some caveats for bNAbs for treatment and prevention. And this is very nicely reviewed in a review article by Marina Kasky in Nature Medicine in 2019. Marina is actually one of my former trainees, and I'm quite proud of her. These bNAbs appear to be very safe. They have minimal adverse events related to administration. And importantly, clinically significant anti-drug antibodies are rare to non-existent. It's pretty clear, as I showed you, that combination therapy is going to be required for treatment, and it looks like 2 antibodies may not be enough. It's also going to be costly. The other question is how can the antibodies be dosed? Can they be given subcutaneously? Because if they can't, then it's going to be hard to deliver and a challenge to the healthcare system. And as far as prevention, it remains unknown whether a single broad and potent bNAb is going to be adequate for prevention, given the variability within the population of viruses of susceptibility to any single bNAb to date. And obviously, there are resistance issues. And the resistance issue requires that we identify the broadest and most potent antibodies to avoid resistance as treatment. And we need to figure out ways to avoid the rapid emergence of resistance. And I think perhaps bNAb therapy may be very similar to the studies that I showed you for IM, cabotegravir, and rilpivirine maintenance therapy, as opposed to de novo therapy. Now, one really exciting thing that emerged in the field, and this is my last part of the talk about monoclonal antibodies, is the ability to engineer trispecific antibodies. And here we have a single molecule that has 3 different antibodies that are capable of binding to the virus in different areas. So this is a cartoon that shows the antibody. Here's VRC01. It's a fragment of VRC01. It's the FAB fragment. CODFV is just a linker that allows you to put the 10E8 version 0.4, which is more soluble than 10E8, in conjunction with an apex binding antibody. So if you could almost think about how it could bind, this binds at the apex, this binds at the emper, and this binds at the CD4 binding site. So if you can picture that initial, if you could picture back what I showed you, you have something on the top, something on the bottom, and something on the side. Some very exciting data. And just to show you here, and these are two SHIVs, DRCO1, this SHIV is susceptible but highly resistant to DRCO1. This one is highly resistant to this apex antibody, but this is highly susceptible. But when you look at the triple, you see high susceptibility. The trispecific antibody has excellent PK, so it's also engineered to be an LS variant. And when tested as prevention in a SHIV challenge model, DRCO1 fails, the apex antibody fails, but the trispecific, no animal breaks through. And this is very, very, very exciting data. And this trispecific bNAb is going into patients, although all clinical trials probably are now on hold until the pandemic is over. I'm now going to finish with islatravir, otherwise known as MK-8591 or EFDA. This is the first nucleoside reverse transcriptase translocation inhibitor. And this is the molecule. It has an ethynyl group, a 3' hydroxy group, which is unique amongst nucleoside reverse transcriptase inhibitors. Remember, like AZT and other drugs don't have a 3' hydroxy group, and therefore its affinity for reverse transcriptase is much lower than this drug. It also has a fluoride here, which reduces reduces the ability of adenosine deaminase to actually deaminate it and metabolize it. So this drug has an enormously long half-life in monkeys and humans. The ethynyl group binds and locks in translocation, which is, as you know, the ratcheting. So after the reading, there's a ratchet movement, and then it goes to the nucleoside site, from the primer site to the end site where the nucleotide gets put in. And this basically freezes things. And then this hydroxy group allows for delayed chain termination as well. So this is an extremely potent, super potent drug. These are data in humans and in monkeys. A single dose of 0.5 to 30 milligrams given here will have antiviral effect that lasts from 7 to 10 days. Single dose. In monkeys, 2 doses, you can see that when you get to 3.9 mg per kg, you can affect a 2 log drop in viral load. This is weekly doses given orally. 2 doses of drug given to these monkeys. And you can see that is very potent antiviral activity as a single agent. So when we started thinking about islatravir for HIV prevention, we first obviously thought about pre-exposure prophylaxis. Here's the high-risk exposure, and we know that HIV prevention is a continuum. There's PrEP before the high-risk exposure, and there's PEP after. So we first did experiments looking at islatravir as PrEP. I'm going to go through this very quickly. If you're interested, my group, I published this with my group in Journal of Infectious Diseases in 2019. Basically, we used a 3.9 mg per kg dose of islatravir. The monkeys were given drug orally once a week. After giving a single dose, they were challenged weekly with 50 TCID of a very pathogenic SHIV, 109C-P3. The control animals were infected, most of them after one week, and another monkey after 2, the last after 4. So this is a very aggressive SHIV, much more so than SHIV 162P3. These are the control animals, and the treated animals were protected 100%. And this was a highly statistically significant result. and with a p-value that's less than 0.00001. Along the same lines, we then waited a couple months because funding became an issue, and then reduced the dose to 1.3 mg per kg. We gave 6 treatments and only 4 challenges because, as I showed you earlier, all the control monkeys were infected by 4 challenges. We waited about 4 weeks, and then we dropped the dose again to 0.43 mg per kg. Again, complete protection, complete protection. Here are the drug levels. We then dropped the dose to 0.1 milligrams per kilogram per week. So just think about that. Average monkey weighs about 8 kilos. So we're giving 0.8 milligrams per week compared to, let's say, TDF, where you're giving 300 milligrams a day. So that's 2.1 grams per week, just the difference in dosing. And you can see that 2 of the monkeys did get infected, but this is a statistically significant protection. And actually, if you count the number of challenges and the times at which the monkeys get infected, we could estimate that the EC90 of islatravir is about 24 femtomoles. I'll show you that in the next slide. Just so you can see that the 0.025 milligram dose did not afford protection and was not statistically significant. So here you can see the MK-859, the islatravir dose. This is the ratio of using 3.9 as the standard. So we reduced the dose by a third. The drug level was reduced by about a third. We reduced that by a third, again, a further third. And then when we got down to the 0.1 mg per kg dose, we couldn't measure drug using the assay, but we could deduce that this was the average concentration of drug. And this is the effective concentration 90 or so, comparable to that seen in the Rhesus macaque with TDF. Why is this important? And here's where we should all get very excited. Data presented by Randy Matthews at the IAS meeting in 2019. Islatravir can be formulated as a soluble polymer, and it is amenable to be formulated in an implant. And here's the implant, very similar to what's used for contraception. And in a study that was presented by Randy Matthews at that same IAS meeting, it was shown very nicely that a 62 milligram implant put into the forearm of a human is capable of giving concentrations of ISL triphosphate intracellularly that are well in excess of what is estimated to be the level of drug needed for protection. So the concept that islatravir may be amenable to implantation for HIV prevention and perhaps in combination with a contraceptive is really exciting. So ISL is completely protective against low-dose rectal challenge. In the Rhesus macaque model with doses as low as 0.3 mg per kg, the EC90 is 24 femtomoles per 10 to the 6 ppmc, and that's very similar to that seen for TDF diphosphate. The half-life in humans is 130 to 210 hours, compared to only 50 hours in Rhesus macaques. And I want you to keep that in mind because we're going to talk about PEP studies in a little bit. And as I told you, it can be formulated in polymeric matrix, and it's very relevant and can be give clinically relevant doses for up to 52 weeks. And remember, implants are removable, and unlike intramuscular injectors, et cetera, that is a really important concept. I'm going to finish up with our evaluation of islatravir as PEP. And I just want to do this very quickly. So we use SIV MAC251 in huge doses. This is a prevention experiment that does not simulate sexual transmission. It's more like exposing an individual to an HIV-infected unit of blood, where 99.999% of individuals would become infected. We gave islatravir at 3.9 mg per kg once a week for 4 weeks. If it was protective, then once a week for 3 weeks, and then once a week for 2 weeks, and then just one dose. All the control animals became infected, obviously, after the challenge. That's not surprising. Oh, by the way, I'm sorry. 24 hours after the challenge, the drug was given orally. And 6 out of 6 treated animals were protected with 4 doses, then 3 doses, then 2 doses. So all 100% protection with 2 weekly doses of islatravir started 24 hours after challenge. When we did the 1 pill, 2 animals did become infected, 1 early on and 1 very late. When we looked at the virus that infected those monkeys, it was wild type. So it wasn't resistant virus. And we think that when you give a massive amount of virus like that, stochastically, there are long-lived cells that can outlast the duration of the drug. And remember, the half-life in monkeys is only 50 hours compared to what it is in humans. But here's data that's generated from Merck and was presented at CROI by Jay Grober this year. If you look at the levels of drug that were protective in monkeys, the 2 doses, this is very easily achievable with a single 60 milligram dose in humans. So here's the minimum PEP protective level, and a 60 milligram dose is well in excess of that. This is a model, but nevertheless, highly suggestive of a very effective post-exposure prophylaxis single agent given within 24 hours of exposure. So in conclusion, advances in daily oral therapy for HIV treatment and prevention have resulted in the availability of conveniently dosed 1 pill once a day options that are extremely well tolerated. Nevertheless, there remains a need for alternatives to daily oral therapy for treatment and prevention. Injectable intramuscular combination therapy is non-inferior to triple combination therapy as maintenance. bNAbs will surely change the treatment and prevention landscape, and implantables are on the horizon for prevention. So I thank you all for your attention. There are too many people to thank for their contributions to this presentation. So I wish you all well. I thank you all for your work. Be safe, stay healthy, and remember the sun will come out tomorrow. Thank you very much.